the material that makes up that membrane, that kind of flat sheet, is the same as the membranes that make up these. Of course, they have different characteristics slightly, but the bulk of it, same. Here's another organelle, membrane bound. Here's extensive membrane, kind of a maze all out here. And mitochondria have membranes inside them. And the cell membrane. It's all the same stuff. And you might as well learn the name for it now. It is, hope you didn't get stopped by a cop. It is a phospholipid bilayer equals and we'll see that coming back a lot. The nucleus has, it looks rather empty in this slide. That's imaginary. Really, the nucleus is packed full of DNA. If I write over here, can you still see it? The nucleus and there's one other little place in the nucleus that you can see shown here. That is what's called the nucleolus. That's Latin for little nucleus. The nucleolus, sometimes people emphasize it, they call it the nucleolus, to emphasize that it's got a few extra letters in there in the middle. Vocab. The nucleolus is just an area within the nucleus. And that is where ribosomes are made. Now, if you're sitting in the back and you can't see my handwriting, please just say something and I'll write it again bigger. So ribosomes are made in the nucleolus. It doesn't have its own membrane boundary on it. It's really just a concentration of stuff within the nucleus. You'll also see that the nucleus, the outer layer of the nuclear membrane, and this is going to be hard to see. So this is one time you have to do. Do you see that most of the nucleus has two of those light blue lines? Two of those light blue lines. Did you all see that? Okay. And you notice that what? Okay. Most of the nucleus has one and then another around it. The nucleus has a double membrane. So it has a single membrane is a bilayer. So the double membrane would end up being 4 of those phospholipid layers. We'll kind of see a close-up of it later. The nucleus has that. And I'm developing this now so that, you see how, right here, and I wish I hadn't had that second cup of coffee this morning. Okay, do you see how there are 2 layers right here? As you, from the inside of the nucleus to out, you have to cross those 2 layers. Do you see that right over here, the outer side of that layer, the whole surface area got bigger, and so kind of pooched out, and it's more of it now. Okay? So the outer membrane of the nucleus in a eukaryote actually gets enlarged and pushed out and becomes this whole structure here, which is the rough endoplasmic reticulum. What? Yeah. Oh, sorry. We don't have players.
Can you try turning off the lights?
Oh no, don't pull the light. We'll all fall asleep and then I'll feel lonely. Oh, you could kill them.
Oh yeah, kill them on the right side or left side and point at one of them. it now.
How's that? That's good. Okay.
I'll
When I was in college, the art history professor thought that he was a great lecturer. And this is before there were lasers. Okay, they hadn't been invented yet. So he always had a big lecture hall and a really, really, really big pointer stick. And one day, in his flamboyant way, he wanted to point to something, and the pointer stick went right through the biggest screen in the biggest lecture hall on campus. And the entire class rose as one inch.
In theory, how many layers are there?
It's really kind of like a sheet. So one phospholipid bilayer actually behaves like a sheet. And the nucleus, in general, has two sheets around it. The Golgi apparatus over here, each little bit has one sheet. Like, sorry? The Golgi, right here. Oh. Yeah. Okay. The rough endoplasmic reticulum is kind of a enlargement. So the outer nuclear membrane.
What's the word after endoplasm?
Reticulum, that's an R, sorry. E-T-I-C-U-L-U-M. Membrane. And why is it rough? Because of the ribosomes. Makes it easy to remember. You've got R for rough endoplasmic reticulum, and you've got R for ribosomes. And what do ribosomes do? Remember? They what? What do they make? They don't make mitochondria. That's mitochondria, my other favorite thing. Okay. What do ribosomes make? Protein, exactly. So, you'd expect to find a whole lot of this labyrinth of membranes. It's a maze of membranes. You'd expect to find a lot of that in a cell that made a lot of protein. And you do. Okay? So we've covered now the nucleus. We've talked about the membrane. We've talked about the rough endoplasmic reticulum, which makes proteins, a lot of proteins. It doesn't make all of the proteins in the cell, but it makes a lot. And then you've also got the outer membrane of the cell. I don't like this picture because it makes it look like the cell membrane has a double layer, like the two double layers of the nucleus. It's just one phospholipid bilayer. That's the outer membrane. What else do I need to tell you about this? What's going to come up later? As far as what you get sick with and where viruses go, we're really going to be talking most of the time about nucleus, rough ER. That's about it. We aren't going to spend a lot of time talking about metabolism in your book.
I have a question kind of what you were saying earlier. So with nucleus, does that mean that the nucleus has 4 layers that are one, or a double layer, that's 4 total, and 2 bilayers?
Okay. Yeah. Okay. I can tell that you're not going to let me details slide. Okay. This is a phospholipid. It has a little roundy part up here that has a positive or negative charge on it, okay? And then coming down from it, it's got 2 little molecules that hate water, okay? So that's your phospholipid. How can you get those? Since things that like water like each other, and things that hate water like each other, okay? So how could you get this to ever be happy in a place where there's water? a lot of water, because there's a lot of water in cells.
Put more of the other.
You have another one facing up to it, so that here's one down here, and these two little hydrophobic legs, they cozy up to each other, and these parts sit. Now, if you want to make a big sheet of this, a big surface area of this, you have all these little heads, and I will attempt to draw a two-dimensional. You get the sense that it goes on forever, that way and this way, and then here's the cross-section of that. And the two little legs. What? Yes! Oh, there's way worse in your body. Okay, so if you look at this sheet now that's got the two sides of it, if you're a water molecule out here, or water in here, you're fine, right? Because the water can cozy up to the polar place where it's happy. But would a water molecule be happy going across the red part, which I drew, which is the hydrophobic, water-hating area? No. It's a little bit like an ice cream sandwich. You know, you've got the bread parts, the cookie parts on the outside. You know, cookies absorb water. They're water-soluble, kind of. But it's really difficult to get any water to go across the really high-fat ice cream layer in the middle. Okay? And if you split an ice cream sandwich in half, and you did a really good job of it, you could split an ice cream sandwich so that half the ice cream went with one cookie on one side and half the ice cream went with the other cookie on the other side. Does this work?
I like that.
Okay, yeah. It doesn't work so well with the Oreo analogy, because when you open up an Oreo, you almost always get the cream on one side or another. Unless you're eating those double stuffs, but those are just bad for you. But if you want to go home this weekend and practice, you know, ice cream sandwich, yeah, you got the ice cream sandwich homework for this course, so you can report back. It won't be extra credit, it'll just be extra weight. But you notice that the thing that you've really got to get here is that this hydrophobic layer is a barrier. Because things just can't, water, ions, sugars, things just can't zoom across the membrane. So the membrane is kind of permeable, but it won't let everything in. It helps to create boundaries. Boundaries equal control. Okay, we all know this, right? So you've got boundaries to keep the inside in and the outside out.
So osmosis doesn't happen in that kind of stuff?
Osmosis will, but only because there's so much water that even a little bit of it crossing will make a difference in the cell. And also, the cells have little doors, little items that are in here that act as portals for different things to get through. It's not a completely closed-off membrane. They're doors to get through. They're quite specific.
When you're talking about that phospholipid bilayer and all that, are you talking about the membrane that encloses the nucleus? Because I was looking in the notes, and it looks like there's something like this for the gram-negative.
Oh yeah, big time. Yeah.
So, but that's on the outside of the cell, right?
Well, it is. This phospholipid bilayer was such a good idea that it gets used many times throughout the cell to do many things. Okay? So you have two complete phospholipid bilayers that are closing off your nucleus, okay, to keep what's outside in the cytoplasm from getting into the nucleus and vice versa. You have to go through the little doors and pass the doorman that's there. You've also got a phospholipid bilayer keeping what's inside the ER from getting to the outside of the ER and vice versa. And you've also got a perfect phospholipid bilayer at the outside of the membrane of the cell. Yes?
molecule in the middle, what is that? Is where is that?
Okay. You have to get really, really small. Okay. And if you were really, really small, this line right here, the dark edge would be the phospho part, the water lichen part, and the light area inside in that sandwich is made up of all the hydrophobic parts of the molecules all clustered together. Is that good? The question is, and I should always repeat questions when people ask them so that you'll know what your colleagues are thinking about. The question was, do you find phospholipids only in membranes? Yes. Mostly. Yeah. That's their primary position because they're involved in the barrier aspect of around the cell. Okay? So let's go back. The membrane-bound organelles that you see that I want you to remember, please remember the mitochondria, of course. The endoplasmic reticulum. Do I need to make you remember the Golgi apparatus? What? Golgi? Yeah. I think I'll put the Golgi in parentheses, so you won't have actually heard about it. But
What does it do?
the Golgi is involved in finishing off proteins, getting them prepared to be released from the cell. And the other one that you have to remember, it's not quite an organelle, but it comes under this heading a lot, is the membrane itself, the cell membrane. You can call it the cell membrane, you can call it the plasma membrane, you can call it the membrane, I'll get there. The Golgi apparatus was discovered by an Italian, Golgi. Italian. So, if you're wondering, do you have to remember anybody's names in this course? You only have to remember the name if something was discovered by an Italian or a woman. That's it. Golgi comes along here. And sadly, there are other great Italian female contributions to science that just don't fall within the scope of this course no matter how much you try so. There you go. Half? Yeah. Oh, okay. My father's family was from Florence. One of those mysteries of the world. Why did my father's family in Florence, Italy?
Someone calls you Sicilian, you get very angry. I gathered everyone else. One of my friends is Sicilian, and one of them is Italian. Maybe it's a big deal that all of them Sicilian. I'm not Sicilian, I'm Italian.
There's a little bit of that. Yeah. Mostly what you get there is a lot of one Italian not speaking to another Italian. Okay. So this is a prokaryotic cell. Now we're going to focus a lot more on the structures and the behaviors of prokaryotic cells. Why? Because these are the ones that make us sick. Okay? So these are the little critters that we need to knock out as much as possible. So I will tell you that everything on this slide, all the words, all the structures, you've got to know. You will be seeing this again on an exam. Probably what you'll see is a diagram with arrows pointing it, and you have to tell me what all the different things are in the book. The reason we need to know this is because as time goes by and we go through the course, almost any part that's named on this diagram is going to be used somehow for us to win the war against bacteria. So, know your enemy. Yes. We've got a flagellum. This is for motion. looking at the things that project out of the cell, there are three. There's a big one, a medium-sized one, and a little one. And they have different names, and they also have different functions. The flagellum is the largest, and it's in motion. Not all bacteria can move, but the ones with flagella can. You've also got the medium-sized one. The medium-sized one is the pilus. The pilus is used for those times when bacteria swap DNA back and forth with each other. And they do this rather indiscriminately. It's a great way for bacteria to swap around disease resistance genes. Antibiotic resistance genes. So the pilus is for conjugation. If I write this over here, can you people over here see how I write? So flagellum, movement, pilus, and the form of this is pili. That's for conjugation. Can you think of a shorter word for conjugation? What's that? Think of a conjugal visit, perhaps. It's for sex. Yes, bacteria have sex with each other. And
What is it saying first?
pili. P-I-L-I is the plural of pilus. Okay. And the last one are the fimbriae. That's the plural. Fimbria is the singular. But I'm not going to quibble if you spell the singular in the middle. The fimbriae are for adhering. Okay? So, one of the deals is, how do bacteria colonize you? They kind of wash up to you, and then how do they hang on? How do they stay there long enough to start dividing and eventually make you sick? A lot of times it's a little fimbriae. Okay? So you've got flagellum, pilus, and the fimbriae are the smallest, and you can see those up there on the top. Inside, well, moving in from here, you've got potentially three layers. The outer layer is a capsule. And a capsule is only on some bacteria and some kind. Sometimes a bacteria will have a capsule when it's growing on one kind of food, but not on another kind of food. Capsules are kind of slippery, slimy. They can be slimy, kind of goopy, and those are actually called a slime layer. If you want to be more specific with the term capsule, that is actually kind of hard. It's firm. Right? Altogether, the capsule and the slime layer, the two types, the larger word for this, the more encompassing word, would be the glycocalyx. Glycocalyx actually means, calyx means covering, and glyco means, say that again? Yeah, it means sweet. So if you ate it, it actually would be kind of sweet. I don't suggest that. Yeah.
So glyco and glucose are the same thing?
They're not exactly the same thing. Glucose, if you use the U in glucose, that kind of implies you're talking about a glucose and you mean a 6-carbon particular molecular structure. But when you say glyco, it's just sweet. So glycolysis works on almost any sugar. Okay. So you have the sweet covering. It can be slimy or goopy. Did you ever leave a glass of milk on the counter and you come back, and as you pour it down the drain, you notice it's become very viscous? A lot like, yeah, I can tell by your faces. Some of you have done this. You've done this experiment without even realizing it. what you saw was the glutinous was the glycocalyx of the bacterial cells that were growing in that milk. Sometimes the calyxes they don't have to have a nasty taste. And some of the food additives that you put into, say, ice cream, make it creamy, the xanthan gum that you put into, that's actually from a bacterium. It's great at making huge amounts of glycocalyx, and they just kind of purify away the glycocalyx and throw away the bacterial cells, and that's where you get xanthan gum from. But you're all going to be checking the labels from now on, and you won't get that. Okay, inside the glycocalyx is capsule, which is the brown layer on this drawing. Inside that, there's the yellow layer. And the yellow layer is the cell wall. Okay? Bacteria, almost all bacteria have a cell wall. And the cell wall gives the bacteria shape, size, determines that their rigidity, and it's made of a molecule called peptidoglycan. Now, I'll only tell you this and make you work through this because if a cell didn't have the hard structure of its cell wall, it would burst. Okay? One of the best antibiotics, one of the first antibiotics, interrupted the process of making the cell wall. That's what penicillin does. So I'm making you learn about the cell wall because if you destroy the cell wall, you destroy the cell. So one more tool in the toolbox. Inside the cell wall, and the cell wall comes in two flavors, which we'll get to later. Inside the cell wall is the cell membrane. And it is a phospholipid bilayer. It's in the light creamy color here. Right there. That's a phospholipid bilayer, just like a regular unit membrane. And that is providing the real barrier in and out of the cell. The cell wall, although it looks like it's really solid here, the cell wall is more like a scaffolding outside a building. You come up to a building and it's got scaffolding all over the front. You can walk through the scaffolding because it's kind of an open network, but you're going to run into a brick wall when you get to the wall of the building. So in cell terms, the cell wall is the scaffolding, and the cell membrane is that impenetrable layer. The only way to get through a wall of a building is to go through the door or the window. So far, so good?
Say that backwards. The cell membrane is the door, and
Yes.
the cell wall is, which is the scaffolding?
The scaffolding is the cell wall.
The wall isn't the wall.
Right.
Okay.
Once you get inside, notice that there are no internal membranes in a prokaryotic cell. There are no compartments. Being in a prokaryotic cell is like living in a studio apartment. Everything happens in the same place. Did you ever live in a studio apartment? Live anywhere? Okay. Right. My husband and I had a studio apartment in New York City when we were first married, and it was fun. You think you know about Inside, in the cytoplasm, you've got the DNA. But the DNA is just sitting there. It's not compartmentalized off. Inside the cell membrane, you've got the cytoplasm. And this also has the ribosomes. And all the metabolism is going on that I'm not going to talk about. I won't talk about it. Inside that, you've also got the DNA. Notice that the DNA, which in the eucs is clustered away, kept safe in that nice, clean little nucleus, in the protein, the DNA is right there. You're dealing with it all. there are also, in addition to the DNA, which in prokaryotes is a long double strand, and it's a circle. It's long. How long? About 4 million letters. You know, letters of DNA, G, A, T, and C. You've got 4 million of these. In addition to the chromosome, and hi, way back there, can you see any of this?
Yeah, a little bit.
Okay. Sorry.
Sorry.
In addition to the DNA that's in the cytoplasm, you'll often have, and this is going to be way down in the corner, so I'm going to spell it out. You also have plasmids. P-L-A-S-M-I-D. Not plasma, but plasmid. Okay? And a plasmid is a little circle of DNA, just a few genes. I don't think they even put one on this drawing. But you'd have another little circle over here. A little circle, like this looks like a big loop of spaghetti. And you'd have another little loop of spaghetti over here. You've got the big circle that's got all the information to make the cell on it, and then you might have a little circle that's got just some specialized information. What's important about the plasmid, because they're little, they're easy to swap. They're like genetic trading cards. One cell can swap and give its plasmid to another cell and get some exchange. The plasmid is often what the bacteria are swapping when they swap DNA, when they're having sex. And why do we care? If it's got antibiotic resistance on it, you've got one cell that's resistant to antibiotics, giving that information, that plan, that recipe, to be resistant to antibiotics to another bacterium. And now you've got 2 antibiotic-resistant bacteria, 2 different species. Back in 1982, when E. coli, you know the E. coli that you get from a badly cooked hamburger that will sometimes kill you? That E. coli strain wasn't around before 1982. It's a newly occurring thing. And the problem was that E. coli must have been having sex with a Shigella. And the Shigella gave it the gene that now E. coli uses to make you really, really sick. And it happened in 1982. You're thinking a few lot of events. There you go. And now you can go on. Okay. Is there anything here that we haven't talked about? You've got basically, this figure is actually quite complex. Because it shows you the loose and goopy slime layer on one side, and then on the other side, it's got the capsule variety of the glycogen. Yes?
What are the actin filaments?
They are very, very kind of basic, simple versions of how to move things around cells. We will never speak of them. You mostly find actin, you think of that in muscle cells of animals and eukaryotic cells. We're not going to talk about this.
The plasma that we use
Exactly. What you brought up yesterday, the plasmid is a way of putting a handy gene into a bacterium, and that's what they did when they got the gene for human insulin, and they put it into bacteria. So now you've got, and they did this in the laboratory. Now you've got a bacterium that can make all the human insulin you want, and that's much better than dragging it out of people. The first gene that was cloned like that was actually growth hormone for people. In the old days, if you needed growth hormone, you got it was extracted from the pituitaries of cadavers. Yep. And I had a friend when I was growing up who entered high school when he was 14, he was 4'1". He was a height of maybe a 5 or 6 year old. And every week he had to go and get a shot of human growth hormone. Every week. They had to find somebody who died and donated their body to science, extract the human growth hormone that was left over in their pituitary, give it to my friend Richard, and during the space of his high school years, he grew a foot to be 5'1". Even though all of his brothers were much taller, they said, sorry, we can't give you any more because there's so many other people that need it. It would be such short supply. Nowadays, it would have been a completely different story. They would have started the therapy earlier, and they would have gotten him to be about the size of his genetic likelihood. Not clear why he stopped making growth hormone.
Plasmids kind of like the tRNA when they kind of swap out.
Ooh, they're nucleic acids, but they have a much different function in the cell. First of all, the tRNAs are RNA, and these are DNA. The tRNAs are involved in an action, making protein. The plasmid would just be the recipe for that protein. Okay? So there we are.
And yes, I wanted to say that we don't really know what mesosomes are. Did you say that? And I totally skipped it.
The question is, so do we not need to know what mesosomes are? No, you don't need to know what mesosomes are.
What about inclusion?
Nah. Sorry. Inclusion bodies are nah. No, that's about it. Okay? Shall we continue? Since these are mostly word sites, I want to turn on the lights. Let's also take a minute and if I pass around the sign-in sheet, can you just sign in? I think I have it here. And, oh, we're doing bookkeeping here. If anyone needs a syllabus, I still have those. Those.